Hello everybody, this is Dr. Ryan from Westboro High School. Today we're going to look at the discovery of DNA as the means to transmit information from one generation to the next in terms of heredity. It was after Mendel's time that we started thinking that chromosomes were likely the molecule that were being carried from one generation to the next because of the way that chromosomes move. They appeared during meiosis to follow along with Mendel's laws of independent assortment and law of segregation. But that didn't tell us exactly which molecule was going to carry the information from one generation to the next, because chromosomes are actually made of two different things, both DNA and proteins. The proteins are histones that the DNA molecules twist around and are actually coiled up around with. So with those two things as possibilities, scientists actually thought that proteins were probably the better option. And the reason is because organisms are so complex that to make a complex organism, you're going to need a complex molecule. DNA was a terribly simple molecule. It's only made of a 5-carbon sugar, some phosphates, and 4 different kinds of nitrogenous bases. Since proteins could be made of 20 different amino acids, and any number of amino acids could hook together in any order that they would like, the proteins are much more variable. So they thought that to make a variable or a complex organism, you needed this much more complex molecule. Well, there was a surprising study or discovery that was actually undertaken by Frederick Griffith, who was studying a type of pneumonia. Now, we know that pneumonia can come from both bacterial and viral causes, but in the bacterial pneumonia that Griffith was studying, there were 2 different forms that he was looking at. One was a smooth colony formed, and the other was a rough colony form. The colony is what the shape of the cluster of bacteria will make when they're grown on a petri plate. Well, what he had discovered is that if you took the smooth colony form, which is shown on the left in the diagram here, that happened to be a lethal form. When it was injected into mice, mice would get sick with pneumonia and they would die as a result of that. Whereas the same species of bacteria also had another variant called a rough colony form. And that R form, when that was injected into mice, the mice would get a little bit sick, but they certainly wouldn't die from the pneumonia. They would get healthy afterwards. And Griffith was trying to figure out why these 2 forms of the same species of bacteria existed. Well, of course, he thought that it was going to be related to the thing that made the smooth colony bacteria smooth. There's a protein coat that is around the outside of them. So he thought that if he killed the bacteria, but he still kept that coat around the outside, that would still kill the mice. So he tried that as an example. He created heat-killed bacteria, injected them into the mice, and surprisingly, the mice survived through that. So now he has a little bit of a problem. It doesn't really make sense to him exactly what it is that's causing this, so he's trying lots of different things. And there was a combination that he tried that was rather surprising altogether. He took 2 safe things. He took the heat-killed S bacteria and the R bacteria, living Rs. He mixed them together, and both of these should have kept the mouse alive, and yet, in combination, they caused the mouse to die. After they performed the autopsy on the mouse, and they took out the bacteria that were inside of its lungs, he found that the bacteria had changed into the S form of bacteria. Something had caused or had allowed the bacteria to be changed from the R form into the S form. And whatever that information was, that was going to be the hereditary material. So something allowed the bacteria to change from avirulent to virulent. That process is termed transformation. And whatever transformed the bacteria was what Griffith knew was going to be the genetic material. Well, there were others who decided that they could study this better. They could try to understand more about which is the genetic material, whether it's protein or whether it's DNA. And there was an experiment that was done by a group of scientists, Avery, McLeod, and McCarty, that seemed to stand out. What they did is they took the heat-killed S bacteria and they extracted it into separate parts. So they had a portion that was protein, they had a portion that was carbohydrates, they had a portion that was lipids, and they had a portion that was DNA. And they took all of those parts and they gave those to the living R bacteria, and then they injected those into mice. Well, when they did this with the proteins, the carbohydrates, and the lipids, the mice survived. However, when they took the DNA portion and they put that into the R bacteria, the mice died. So that seemed pretty well to say that DNA was the transforming material. But not all the scientists agreed. And the reason that they said that it might not be exactly telling was because they thought the DNA would be rather challenging to be able to extract from those bacteria and to have it just pure DNA. So they said that there was probably some protein that was mixed in with that, because they knew that the chromosomes were made of both DNA and protein. So they thought that that wasn't conclusive enough. So they thought that there should be more experiments. And there was. So Hershey and Chase decided that they would conduct an experiment also. In this case, they were experimenting with a type of virus. Now, we haven't had a chance to talk at all about viruses during the year, but viruses are non-living, obligate, intracellular parasites. What that means is that they have to use another organism's cells to be able to reproduce. They inject their genetic material into the cell, and they take over the machinery of that cell to be able to cause it to make new virus particles that would then burst out of this new cell. So this organism this thing that we're going to work with is called a bacteriophage, or you can just refer to it as a phage. And as you see in the diagram on the left, it's composed of two different things, DNA and protein. So what Hershey and Chase decided that they would do is they took radioactive phosphorus, phosphorus-32, and they allowed the viruses to grow in that radioactive phosphorus. The reason for growing it in that is because phosphorus is present in DNA, but it's not found in proteins at all. So by growing these viruses in this radioactive phosphorus, the DNA would be labeled with this radiophosphorus tag. They then took that phage, they put it in with the bacteria, they allowed it to infect the bacteria, then they took that and put it into a blender. And the reason inside of a blender is that it would shake loose the particles, the phage particles, that are on the outside of the bacterium. They then put it into a centrifuge that spins really fast, and the bacteria, being significantly larger than the phage would be, the bacteria moved to the bottom of the test tube. They could then test and see whether the bottom of the test tube, called the pellet, or the liquid portion above it, called the supernatant, was radioactive. And when they did this with the radioactive phosphorus, they found that the inside of the bacterium had become radioactive. It was the phosphorus part. So they completed the experiment a second time. The second time, they made radioactive sulfur. The sulfur is not found in DNA, but it is found in proteins. And they then did the same exact test. They allowed the virus to infect the bacteria. They used a blender to be able to shake it loose. They used a centrifuge to get it to separate into a pellet and the supernatant liquid. And this time, the supernatant liquid, the part that housed the virus particles, was the part that was radioactive. The bacteria were not. This convinced scientists that it was DNA that was the transforming bacteria. It was convincing proof that DNA was the genetic material. So we still have this problem, though, about understanding what the structure of DNA is. Prior to the study that Hershey and Chase had done, Erwin Chargaff had been looking at the structure of nucleic acids, and he actually had found two rules that were rather important in helping understand the structure of DNA a little bit later on. One of the things that he had found out is that with respect to the nitrogenous bases, the percentage of adenine inside of an organism was very close to the percentage of thymine, and the percentage of guanine was very close to the percentage of cytosine. So that's a rule that Chargaff came up with, that the amount of A is equal to the amount of T, and the amount of G is equal to the amount of C. One of the other things that Chargaff had discovered is that the relative amounts of adenine, thymine, or guanine, cytosine in one species were not the same as what they would be in other species. So he thought that this could be the hereditary material. Remember, this was prior to Hershey and Chase. He thought that because it meant that this 4-nucleotide or this 4-base nucleic acid was a lot more complex than what they had originally thought. So there were other people who also did work to help understand what the structure of nucleic acids were. One of those people is Rosalind Franklin. Rosalind Franklin used a tool called X-ray crystallography. And what she did with this is she would take a form of the DNA and she would strike that with X-rays. X-rays were discovered in the early 1900s, so this was a relatively new science. And then by the way that the X-rays would diffract, they would bend ever so slightly, they would deduce what the structure of the molecule was that they were taking the picture of. She happened to take one especially telling photo that was named Photo 51. And Photo 51 is the photo that actually was passed on to James Watson and Francis Crick, the two people that I'm going to talk about next. Watson and Crick used Photo 51 to build a model of DNA. This was without Rosalind Franklin's knowledge that they had used that information, but once they figured out the structure of DNA, they published an article in Nature magazine that described how DNA was put together, and they were then awarded the Nobel Prize for Watson and Crick, along with a collaborator of Rosalind Franklin, Maurice Wilkins, were awarded the Nobel Prize for coming up with the structure of DNA.